Windows to Mac computer. So that's been an adventure. And it's also been a while since I used Zoom because it seems like most of my contacts use uh, different conferencing software now, particularly Teams. Anyway, glad to be back on Zoom.
This is how it goes, Chris.
And I'll tell you a little bit about mollusk first. I know you've got a lot of birders, maybe some flower people, a lot of photographers, might not know too much about mollusks. Just to remind you that this is the second largest phylum, second most diverse phylum, I should say, of animals after the arthropods. And it's got a really wide variety of body plans, everything from snails and clams through squids, which is about as diverse as you can get. And I'm going to be talking about bivalves today, but these are the two big classes in the phylum. The gastropods are the most diverse, the bivalves second most diverse. And I always feel like making a pitch for mollusks. They amount to about 8% of animal biomass, which is a remarkable fact. And humans amount for about 2.5% of animal biomass. So we are outnumbered and outweighed quite a lot by the mollusks. There's been some malacologists, that's people that study mollusks with Wigness. Probably the first one was Leslie Hubricht, and I don't think any of you folks are likely old enough to remember Leslie. I never met him, but he moved to St. Louis in 1923 and got hooked up with Wigness in the very early days. And there's been a couple of biographies of this guy, and it's said that Wigness is probably largely responsible for him becoming a really productive amateur scientist. He published a lot, including his magnum opus, which is this compendium of land snail distributions in the eastern United States. And it's a very important work. And here's a couple of other folks that some of you might remember if you're old enough. Ron Ash is a really prominent Missouri amateur biologist who published a lot. He published field guides and gathered lots of distributional information about crayfish and snails and bivalves. And when I came to Missouri and first started working on freshwater mussels, his book, Missouri Naiads, was a really important resource for me. And his friend here, Frida Schilling, was also an amateur malacologist. She and her sister did a lot of collecting in the rivers in eastern Missouri back in the day, and their collections are now in the Field Museum. Anyway, I got to know Ron and worked with him on a book about the land snails and slugs of Missouri that was published by the Conservation Department a few years ago. And this is available online through the Conservation Department or through BearWorks at missouri-state.edu. But again, Missouri Naiads was a really important resource for me. There's also it's sort of an updated guide to freshwater mussels, which is the main topic this evening. And this is also available on BearWorks, or I think from the Conservation Department. You can probably still get paper copies of this. I don't think there ever have been paper copies of the snails book. Anyway, we're going to talk about one of those 7 classes. This is the bivalves, about 10,000 living species. And they're important animals for a lot of reasons. They're all aquatic. That's kind of limiting if you're earthbound, like many of us are. But they're very abundant and relatively large body. Many are important as food, as you know. And as we'll see, their ecological impacts extend into freshwater as well. About 12% of these species live in freshwater, over 1,000 species worldwide. The key features of the bivalves, just to get you oriented, are number one, that they're bivalved. That is, they have 2 shells. And you know this, that the shells are hinged at the top by a dorsal ligament. And there are apertures at the back end of the animal that direct water flow. These are suspension feeding animals. So water comes in through an in-current aperture, passes through the gills inside, and then goes out through an excurrent aperture. They've got a muscular foot, which is kind of like a tongue. It's got no hard parts, but it's very muscular, and it's got hydraulic spaces in it, so it can be extended with hydraulics and retracted with muscles. And they use this for crawling and burrowing. And then they're capable of secreting protein threads called byssus that can attach them to a hard surface. This isn't terribly important in our native mussels, but the non-native zebra mussel, which is a very different critter, attaches to surfaces, and byssus is a big reason why it can do that. So if you take that shell off, you'll see that inside there are these paired gills on both sides of the animal, and these are hollow structures. They're ciliary filtering mechanisms, and they drive the water flow. There's also a well-developed digestive tract. Food is passed to the mouth at this end, and it comes out the back and shoots out with the excurrent water flow. Now, it's hard to get oriented on an animal that doesn't have any eyes. If they had a head, which they don't, they would be face down. Typically, native mussels are infaunal. They're at least half buried and often essentially completely buried in the substrate. And they use the foot for burrowing. They can extend it and then spread the end of it like an anchor and then pull themselves down. And they're very adept at burrowing. I've got a video clip here that is sped up. This is about 10 times speed. And if you watch this foot, it's remarkably agile, and you'll see that it's capable of picking up the substrate. There are cilia all over the surface of it, and there's mucus being secreted, and that mucus is adhesive. So that's what's adhering to the sand, giving the animal some purchase too when it eventually inserts its foot into the sand, flips itself upright, and then pulls itself down quickly. And you can see the two apertures here, so you know that this is the posterior end of the animal. And here he goes. Now, this is not a native mussel. This is an Asian clam. I'll tell you a little bit more about the differences between these shortly. And here's a video of some very young native mussels. And you'll see that they are also quite agile. And they look almost more like snails than like clams at this age. And they're very active. And at this stage, burrowing is kind of moot because the substrate particles are as big as they are. These are literally the size of sand grains. They're that small. So burrowing is more like crawling through boulders for them. Adult freshwater mussels aren't nearly as active as that. They're not like the coquina clams that you might see at the beach in Florida. They tend to get themselves into position and then they'll stay there, either for days or sometimes for years or even decades at a time. People have marked adult mussels and come back to the same site 10 years later and found that animal in the same spot. Now, you don't see the mussels here. They're not real obvious, but you see these pairs of apertures. And these three mussels, that's all that's showing. So they're able to access the water, and that's all they need. Now, if they wanted to come out, they could, or they could dig down further. But this is their preferred position. They're safe, but they've got access to the water for food. Even though mussels look like they're not doing anything, they're usually actually very busy and have relatively high metabolic rates for an invertebrate. This is an endangered species, actually. This is the winged maple leaf. And I'm going to stick some food color in front of its in-current aperture, and it will draw that food coloring in. Now it's in the mantle cavity, it's entering the hollow gills, and then it's shooting out the excurrent aperture. The in-current aperture is kind of frilly here. It's a filter, and the excurrent aperture is smooth and nice rocket nozzle for the exhaust. Here's a much smaller mussel, and we've got some algae in the water now so that you can visualize this filtration better. And there goes the fecal pellet. But you can see water isn't entering just through a posterior aperture at this stage. They don't really have well-developed apertures because, after all, they're embedded in the substrate completely at this stage, and there's plenty of room, as it were. So they're taking water in all through the gape of the shell, anterior, ventral, and posterior as well. But the excurrent aperture is important. It's still shooting that water out the back. I hope I'm not boring you with this anatomy, but it's hard to relate to a mussel and the things they do if you don't know how they're built. So here's the shell, and I'm sure you know that they've got these nice growth lines, and those growth lines are actually just like tree rings. They grow from the edge of the shell, and you can determine the mussel's age by counting these lines. And you can tell when it had good years and bad years by looking at the width of these growth lines. It's not quite as clean as tree rings because there are other things that can influence their growth, disturbance, changes in water quality, and so on. But you can often get a pretty tight read on age. This individual from the Sac River was about 40 years old when we picked it up in 2008. And we've done quite a lot of work in the Sac below Stockton Dam, and it's pretty clear that washboard mussels, this species, haven't recruited in the Sac since Stockton Dam closed in 1970. That's a long time ago. You can still find them. Washboard can live as much as a century, but they're becoming more and more rare and eventually will be extirpated there. Operation of the dam is apparently inimical to their reproduction. Let's take the shell off and look at the anatomy some more. Here's the mouth and the foot. The two valves of the shell are closed by adductor muscles. You might remember this if you're my age. You probably dissected a freshwater mussel in freshman biology. There's the apertures here at the back end. And now what we're going to do is get you oriented by putting some eyes on this mussel. Now, mussels don't have eyes, but if they did, there you go. Now you understand that this is the anterior and the head end, and they've got the one muscular foot and then the apertures at the back. Now we're going to cut them in half and open them up. And now you can see these are from a classic invertebrate zoology text by Pearson Bushbaum. Of course, I've modified them a bit, but the basic black and white structure is from Pearson Bushbaum. The gills are hollow and they're perforated. There's lots of little openings. We'll call them pores. The water comes in the back end, and we know now that it can come in anywhere along the gape of the shell, passes into this cavity inside the shell. The inside of the shell is called the mantle cavity. And then it passes into the gills through the pores and moves in vertical spaces called water tubes to get up to a dorsal water passage. And from there goes out the excurrent aperture. Food particles get trapped on the outside of the gill, and they're transported by, again, ciliary mechanisms like the cilia that line your airways and your lungs. And then the food reaches the edge of the gill and gets passed in a mucus string up to the mouth. Very mucousy animals. Okay, now remember your anatomy lesson as we go on. Little bit of taxonomy here, just very little. In freshwater, we've got the pearly mussels, which is what I'm talking about tonight. These are called the Unionida or the unionid mussels. This is an order of bivalve mussels, an order of the class of the phylum. And we've got about 300 species of these in North America, north of Mexico. And their life cycle is what sets them apart. They're really dramatically different from any of the other bivalves and really from any other animal on Earth in the way they do business. I'll tell you more about that. We do have quite a few other bivalves in freshwater. We've got our native fingernail clams, 40 species of those in North America. They're little tiny guys. They don't have the complicated life cycle. and they don't get nearly the attention that they should, but they're actually quite common, reasonably abundant. You can find them in ponds, which is remarkable. How do they get to a pond? Darwin found out that they ride on birds. That's a pretty interesting trick. And then we've got Corbicula, actually 3 species of Corbicula now. This is a non-native genus of freshwater bivalves, and we call them clams to distinguish them from mussels. And then I'm sure you're aware that we've got some other invasive species, the zebra and the quagga mussels. Corbicula, we think they're causing some problems, but they're not nearly as bad as Dreissena, the zebra mussels. Zebra mussels are bad because they attach to surfaces and they become very common, and they can block pipes and weigh down boats and docks, and they're just a nuisance that's a very expensive nuisance too. So we're talking about unionids. Unionids are good. The Corbicula and the Dreissena, well, they've got advantages and disadvantages, as we'll see. Here's the deal with the native mussels, the pearly mussels. They've got this bizarre life cycle. The adult mussels can live for decades, even a century or more.
Hey, Pat.
And they produce a very large number of very small larvae. Larvae are called glochidia. Glochidia are parasites. They have to attach to the gills of particular species of fish. And there they get encapsulated on the fish's gill, and they undergo a metamorphosis. They don't grow so much. So some do, but most of them don't. Only the very smallest ones grow, and they only grow to be the size of a normal-sized larval mussel. So they don't really take much from the fish. So they don't do much harm as long as there aren't too many of them. But they undergo this metamorphosis to the juvenile stage, and then they drop off the fish and grow up to be adult mussels. So the question is, how do the glochidia get to the fish? And this is the answer. This is a fairly common mussel in Ozark streams, and in the tributaries of the Merrimack and the Big River, you've got some similar mussels. It's called broken rays mussel, Lampsilis reviana. And this is the posterior end of the mussel. And what looks like a little fish is actually a lure that this mussel is displaying to try to attract its host fish. You can also see a structure here like a sort of a pleated balloon, and that is the gills of the mussel. And she's got her larvae tucked away inside that gill, and she's putting it up there in harm's way so that when a bass or a walleye comes down and strikes at that lure, she'll get her larvae onto the fish. So the marsupial gill is interesting. Here's a representation of the ovary of the female mussel. When she lays her eggs, they're transported into the water tubes in the outer pair of gills, and these are the marsupial gills. So that's what you're seeing right here, this pleated area. And here, that's what you see here. And they make a lot of larvae. Parasitic life cycle is, kind of a low odds of success, so they have to make a lot of larvae, and up to a third of their body weight in some cases. The little babies are kind of busy in there. Once they're developed, they start exercising, waiting for their opportunity, which is singular and brief. They only get one chance to get onto a fish. And here is a dramatization of the process. The fish strikes the marsupial gill, breaks it open, and then inhales a very small number of the larvae compared to the number that it dumped into the water. They clamp onto the fish's gill and become encapsulated, and they live as parasites for a couple of weeks, maybe a month. The fish's tissue grows around them, protects them, nourishes them until they've completed their metamorphosis to the juvenile stage. So that's a pretty good trick, to have a fishy lure on the back. And there's a wide variety of these things that have developed in in one of the groups called the Lampsilinae. This is one that fishes for walleye. This is one that fishes at night for rock bass. These are fishing for smallmouth and rock bass. Those are the mantle lures. Mussels can also release what are called conglutinates, and these are aggregates of eggs containing larvae that act as baits. And they're mimetic. They're dressed up to look like a worm, or, as we'll see, blackfly larvae, or even small fish on a transparent fishing line that is a mucus cord that the female secretes. So this thing is floating around in the water. Some of them dispense their conglutinates in response to the touch of a fish that approaches the mantle. Others actually grab onto the fish and hang onto it and blow their larvae into its mouth. Now, this is the kind of stuff that got me really interested in mussels. It's just charming. Here's conglutinates again. Again, these are their eggs all stuck together, formed in the water tubes of the female gill, molded, if you will, and then dumped out into the water to be bait for the fish. And this is a Tychobranchus mussel, kidney shell. And these are its little conglutinates that are being released from the female. This is the posterior end of the female. And this is the host. This is a rainbow dart. No, actually, this is orange throat darter. But most darters can serve as hosts. The conglutinates are pretty clearly decorated to look like something, and what they seem to be mimicking is blackfly larvae in the case of our Ouachita kidney shell. And then there is a related species called the fluted kidney shell, and its little conglutinates mimic the pupal stage of blackfly. And I think you'll agree that these are elegant, but they're just bags of glochidia, and what seem to be eyes are just painted on, and these are weak spots. So if the fish bites this thing and doesn't actually rupture it, the glochidia spill out of these fake eyes. This isn't a very high resolution video, but you'll get the idea. The conglutinates have an adhesive tail that sticks to the rock, and the darters come by and say, yeah, that looks like something tasty. You can see the glochidia puffing out from under its gill covers. I guess it's kind of a spicy food. And they almost always spit the thing out after they chew it up, but then they go back for another one. So they don't actually swallow very many glochidia at all, but they do disperse them into the water. And of course, a fair number of them will attach to the fish's gills. But I think the coolest adaptation of all is in the genus Epioblasma. They've got a lure too, and when the fish investigates the lure, instead of being bitten, the mussel grabs the fish. We used to have a couple of these in Missouri. One of them is extinct now, but we still have one. You see this tiny little lure. It's like two little twiddly fingers. It's a great lure for a darter or sculpin. Missed. The fish wise up pretty quick. Darters are remarkably personable fish. So this fish has triggered this thing once, and now he's back to do it again. He knows something's up, so he's just messing with it. What you need is a young and naive fish, like this one. He's probably never seen this before, and he's so small that he falls into the gate. Now, the mussel feels the struggles of the fish, and it'll hang on to it basically until the fish stops struggling, and it's nearly suffocated. And all the while, it's releasing glochidia into the mantle cavity in this case, and a lot of these are attaching not just to the gills of the fish, but also to its skin. And when this fish struggles, it's going to disperse some glochidia into the water, and other fish that come to investigate, see what's going on, they're going to get infected with glochidia too. It's really hard on the fish that gets captured. I like to think of it more as using that fish as live bait. And I expect that because darters are fairly social and they do come in and investigate, that the real target is the looky-loos, Okay, so they're charming or terrible, depending on how you feel about them grabbing fish by the head. Um, are they important? And I would argue that yes, they're important in three major ways. One is through their feeding. And I'll detail this. They feed very low on the food chain. They feed on stuff that's suspended in the water. And that's algae, bacteria, the breakdown products of leaves. You know, what do leaves turn into when they fall in the river? Well, eventually they get broken down into just particles of cellulose encrusted with fungi and bacteria. And when that stuff gets small enough, it's accessible to mussels. They also have economic significance, as we'll see. And the reason that I spent most of my career studying them is because of their conservation significance. So what about this suspension feeding? Now, I was kind of equivocal about our corbicula and zebra mussel because in spite of the fact that they cause some problems, they do this part of the bivalve story just fine. In fact, this video clip here is of corbicula. And what we're going to see is me polluting the water here. I'm going to pour a bunch of algae into the water. And now we're going to speed this up so that we don't have to wait for hours to see anything happen. And what you're going to see is a lot of turbulence. There's a bunch of little corbicula clams on the bottom of the beaker here, and they're filtering the water, which is what bivalves do. So you can see the turbulence that they're producing. And you can also see some chunky stuff floating around, and that's biodeposits. That's feces and pseudofeces released by these corbicula clams. You can see their apertures there too. Pseudofeces, right? Now that's kind of silly. But pseudofeces is particles that they filtered, but they didn't swallow. If there's too much stuff in the water for them to ingest, they'll keep right on filtering, but instead of swallowing all of it, they'll be more selective, and a lot of it they'll just dump off the edge of the gill, bound up in mucus, so it's particulate, and that's what's called pseudofeces. And not just native mussels do this, but zebra mussels, corbicula clams, all bivalves do this. And this is food for other organisms. Both the feces and the pseudofeces of mussels are perfectly good food for aquatic insects, for some fish, and for annelids. So they take stuff out of the water column, algae, bacteria, even sediments, you know, mineral sediments, they'll filter that out too. They dump it out as waste on the bottom of the river or the lake. Nutrients are released back into the water, and then benthic organisms, aquatic insects, snails, fish, it's food for them. This is a really neat study that was done quite a while back now, but these researchers had cages and they put fish in the cages. The fish they used were larval lampreys. You think of lampreys as bloodsuckers, but a lot of them are just deposit feeders, kind of like chordate earthworms. They feed on organic material. And they had some cages with just lampreys and some cages where they put a mussel in as well. And over 80 days, these little larval lampreys, which are fish, grew twice as fast if they had a mussel in the cage with them. And of course, the reason for that is the mussels were producing their biodeposits and nourishing the lampreys, which grew twice as fast as if they were alone. And then, of course, the mussels themselves are food too for other organisms. There's quite a few mussels that are taken by raccoons and muskrat and otter. Fur-bearing mammals are hungry for mussels, especially in the wintertime. There are certain fish that specialize on mollusks. Red-eared sunfish eat snails and bivalves too. Freshwater drum. Big catfish sometimes feel like a bag of rocks if they've been feeding on mussels. They'll eat adult mussels, just swallow them whole. I don't know how they deal with the shells, but I guess they manage. Crayfish and people, not currently, although I got into an argument with a guy on YouTube just the other day who was claiming that he ate freshwater mussels all the time and how they were really delicious. And he said he'd eaten 30 different kinds. And I had to point out that the state that he's living in, it's illegal to harvest mussels for food or for bait. But anyway, and for you bird people, there's even a bird that eats mussels. You're probably aware, if you're a birder, that limpkins are showing up in the Ozarks now. And yeah, they eat snails and they'll also eat freshwater mussels. If you're paying attention and you look inside places where muskrat or an otter might hang out, like this is a hollow log that Melissa is sitting on, and she's pulled all these empty shells of mussels out of this log. They didn't crawl in there. They were eaten by probably by a muskrat. And this is what you call a midden pile. So that's their ecological connection. They're a link in the food chain. Their economic significance has to do with shells and shell industries. Native Americans harvested mussels for food in the winter, but they were probably starvation food. They're not particularly nutritious so far as human consumption goes, but they used them a lot as tools. The maize farmers along the Mississippi used shell hoes, big washboard shells hafted onto a wooden shaft, and then use that for hoeing your garden. Tools like scrapers for pottery. If you find an old mussel shell with a beveled edge like this, it was when it was used as a scraper for pottery, and of course ornamentation. People have been using the mother of pearl, the nacre, for ornamentation probably since before we were really human. This is some artifacts in a museum in New Zealand. The Maori harvested freshwater mussels from lakes, and they used a dredge. And this is the remains of a Maori dredge, probably 200, 250 years old, that was used for mussel harvest. In modern times, though, it's the ornamentation thing that has been important. A German named Beppel arrived in the Midwest in the 1880s, and he had been a button maker back in Europe, and he realized that freshwater mussel shells were a valuable resource for this purpose. And he started an industry that took off like a rocket and became what would in modern terms be a multi-million or a multi-billion-dollar industry that lasted for well, a good long time, till about 1948, when plastic replaced pearl and when the button fishery, the shell fishery, crashed as well. From about 1952 on, the Japanese started an industry using beads of freshwater mussel shell as the nuclei for marine pearl oyster culture. And that is still going on today. It's the best material for nucleating a cultured pearl in a marine mussel. And then even more recently, as we'll see, freshwater mussels have become important in pearl production. But back to the button harvest, these are shells piled up at a site near Fairport, Iowa, back in the day. And this was not a harvest so much as a mining operation. Mussels were extremely abundant, and they were harvested in a non-sustainable fashion, let's say. And factories went up and down the river. Even as far west as Iola, Kansas, there was button harvest and a blank manufacture operation. I think the finished buttons were produced in the factories along the Mississippi, but the blanks were prepared as far west as Iola, Kansas. If you've got your grandmother's button box, you've probably got shell buttons. You can still find these online, some of them very decorative. There's actually a National Pearl Button Museum in Muscatine, Iowa, that's worth a visit. And then the shell bead thing, well, again, that's still going on. The very most valuable marine pearls are nucleated with freshwater mussel shell. But the average pearl that you're going to buy if you're not wealthy. This strand is a beautiful strand, cost $16 a couple of years ago. Well, those are freshwater mussels, freshwater shells, I should say. They are able to nucleate freshwater mussels in China, and this has become the primary source. I thought I had some slides of this, but I guess I don't. Anyway, let's move on to conservation before I run out of time here. Mussels are in trouble, and this was a fact that was clear at the end of the button days because the harvest really declined rapidly, partly from overharvest, probably largely from overharvest, but also because of pollution. And today we find ourselves in the situation that of roughly 300 described North American species of unionid mussels, our native pearly mussels. At least 34 species are extinct, and 88, now I believe this is actually up to 91 species, are listed as federally endangered or threatened. And these are our endangered species here in Missouri. And you can see they're beautiful animals and quite diverse. And all of these are on the rocks, so to speak. In this day and age, it's not because of overharvest, it's because of habitat destruction. There's lots of reasons for mussels being endangered. And of course, their significance changes as they become less abundant. I've argued that they have ecological and economic importance. Well, when mussels become less abundant, that importance goes down and down and down as they become rare. But if you get rare enough and you're in danger of extinction, well, then your conservation importance goes way up because we have laws to protect species that are endangered. This is a pile of button shells from a site on the Ohio River. It was actually a piece of a sidewalk that was being cut up. These shells, they were just hugely wasteful. You know, they would cut out as little as maybe one button blank toward the end of the harvest, and then they would just dump all this shell. They used it partly for animal feed, but most of it just ended up being used as fill or just dumped. I understand that any place you dig a hole in muscatine, you're going to hit button shells like this because, I mean, they raise the surface of the land like a tell in archaeology with all the waste shell. This always reminds me of the Pol Pot era. Remember when they gathered all the skulls up to display the people that had been killed in Cambodia? Well, certainly the button industry did a number, but the current declines, the ongoing declines, and the fact that mussels haven't recovered their abundance, there's been 45 studies that were put together and meta study done by Dave Strayer a few years ago. And the most commonly cited cause is water pollution, water quality degradation. And close behind that is habitat destruction, which is mainly dams and impoundments. Mussels are most abundant in the big rivers. And of course, it's the big rivers that get the big dams. And impoundments are not good for mussels. It changes the fish fauna. They've got to have the right fish to reproduce. And it also changes the habitat in ways, no more moving water, lack of oxygen on the bottom of big reservoirs and so on. So lots of things going on. And the mussels have to blame themselves to some extent because they've got this weird life history. The parasitic larval stage, you've got to have the right kind of fish, and you've got to have them at the right time, and you've got to have them in the right abundance. All of these are changed by channelization and damming of rivers and siltation, introduction of non-native fish. All of these things interfere. The adults, they've got their requirements too for stable benthic habitats, and the juveniles even more so. They're susceptible to being washed out in a flood, to being silted in and buried. They're fed on by micropredators that may become more abundant in enriched systems, and they're very susceptible to pollutants. They're intimately exposed to waterborne pollutants. So sewage, dams and impoundments, erosion and siltation, and even insult to injury, non-native bivalves like zebra mussels will attach to native mussels and keep them from moving and feeding normally. Well, we just passed the 50th anniversary of the Endangered Species Act, which, as you know, was implemented back in the Nixon administration. Amazing. To protect how Republicans have changed. To protect animals and plants from extinction that was resulting from economic growth and development. And it's supposed to protect habitat of endangered species. And it's supposed to apply regardless of how economically important a particular species is. And we also had the Clean Water Act in 1972, which set the EPA the task of establishing water quality criteria, which are the allowable limits for important pollutants. So ammonia is probably the most important water pollutant, and there is an allowable limit. It's kind of a complicated one because it depends on the pH and it depends on the temperature, but there are water quality criteria for ammonia. And if a stretch of river is impaired, then powers that be are supposed to do something to fix that up if they're following the EPA established guidelines. How do you determine these guidelines? Well, they're based on the sensitivity of aquatic organisms, and this is established by laboratory scientists that do toxicology research. And I got tagged by these folks way back when because I was raising freshwater mussels for restoration projects, and they needed little mussels to do toxicology on. So this was a match that's worked out very well over the years, and I'm still working on that even in retirement. Involved in a couple of projects with folks in Colombia that do mussel toxicology. The way this works is the water quality criteria are based on the most sensitive species determined by research in at least 6 categories. And these are categories like a salmonid fish, not an issue in Missouri, but in the West, it's a big issue. The second fish family, a chordate of some kind, an aquatic chordate, and that could be a lamprey, it could be a salamander, it could be a finfish, and so on it goes. And there's 2 categories where a mollusk can fit in. So it turns out that a mollusk can actually have a lot of impact on water quality criteria. And it turns out, too, that mussels are very sensitive to some pollutants. They're in the top 5 for certain pesticides like allichlor, for ammonia, which is huge, potassium chloride, sulfate, nickel. And they're in the top 10 for sodium chloride. That's table salt. Is that a pollutant? Well, you bet it is, because it's used for melting the ice in the wintertime. And the concentration of sodium chloride, particularly in northern rivers, is just out of sight. And it's becoming an issue. There's been a lot of research on this in Canada in particular, not surprisingly. Copper. Anyway, if you do the research and find species that are more sensitive and present that data, and if those data are respectable and they're accepted, you can alter the water quality criteria, make them more protective. So this has been ongoing for a long time. These are some of my colleagues at the USGS CERC, the Columbia Environmental Research Center. This is a group of aquatic toxicologists, and they've been working on mussels now for 20 years, among other things. And our big accomplishment in that time was to get the EPA to lower the ammonia criterion. And they lowered it a lot. They lowered it by half in 2013. Unfortunately, they lowered it so much that it's become problematic for a lot of folks to actually meet the guideline. And it is being met in some places, but not in others. Still, it's a good thing because ammonia is a very bad pollutant, and it's not just bad for freshwater mussels. It's bad for any aquatic organism. Mussels in the field can also be important, not just in the laboratory. They're not that hard to find if you know where to look, and their presence is a good indicator of water quality and an intact fish community. So you can use it to identify parts of a river that are in good shape and need to be protected. And in this sense, they're actually very useful because they live a long time. They not only mean it's a good spot, but it's been a good spot. Other organisms that are used for this that are more ephemeral, like aquatic insects, don't give you that long-term evidence. They're also used for biomonitoring. We've developed caging techniques for freshwater mussels, and marine mussels for decades have been used for contaminant monitoring. You put them out in a cage, you see if they survive, but you can also collect them again, and you can analyze their bodies and see what the concentrations of pollutants are by measuring their body burden. So mussels have become important for natural resource damage assessment. And this is a government program, federal program that identifies areas that need to be restored and punishes polluters by making them pay for the restoration. And mussels are very useful for this because if you have a mussel kill, the bodies don't just wash away. Well, the bodies do, but the shells are hard and they can last for centuries. So they can provide good evidence of a kill. Trouble is, it takes them a decade to come back in that spot once they're gone. And there have been some big assessments. I don't have any current ones. I really need to update this slide, but the Asarco settlement in 2009 was $65 million in damages, not just for mussels, but mussels played a big part in this. Oh, I've got yuan in here because I gave this talk to some Chinese folks too. 2010 Doe Run settlement, $72 million in damages. And yeah, it's an issue. So are mussels significant? Yes, they are. And they're an important part of rivers. Even if they seem obscure, they're tied in with everything going on in rivers. They're a link at the very base of the food chain. So they make, and you know, rivers they're not autotrophic organisms, so to speak. A river doesn't generate enough algae to feed the animals in the river. Rivers feed on terrestrial detritus. We say that they're heterotrophic organisms. A river eats dead leaves. It eats organic material that's coming in from the land. And what digests that is the mussels and the aquatic insects that capture that detritus, break it down, and turn it into something that's more palatable for other animals. Mussels have economic value historically and presently, and they can improve water quality in terms of just filtering the water if they're abundant enough. And of course, endangered species are an early warning system. And, you know, you can look at mussels in this way and say that they have value because they contribute to these human interests, but it's also important to note that these things are irreplaceable. They're beautiful and they're wonderful. And, you know, even in the relatively short time that I've been working on them, which amounts to about 30 years now, I've seen them going away, and it's a sad thing. And that's why I like to give talks like this, is to make more people aware. And I will stop. And thank you for not interrupting me, but I do hope that you have some questions.
Chris, thank you so much. I think that was absolutely fascinating and informative. We really glad we had you here or had you with us tonight. Appreciate that. Thank you.
Likewise. I've just opened the chat. I don't see any questions on the chat.
Okay. Any questions in the room? Dan.
So, I didn't hear how they reproduce. Are there male and female mussels?
Bill, you're going to have to repeat the questions for me.
Sure. The question was about how they reproduce. You know, they're both two sexes. How do they go about that?
Oh, that's an interesting question. I was going to go into that, and then I decided that it might be a bridge too far. But if you're really interested, I probably don't have to go too far forward in my slides to come up with that. Okay, here's how the boys do it. They release structures called spermatozoomata into the
Chris, I
water.
think you might have stopped sharing.
Okay, good point. Okay, can you see him now?
Yes, we do.
All right. This is a male mussel, and that is not going to play. All right, hang on just a second. Should have spent more time getting my slides organized. I can see that. This one should work. Yeah. Now tell me, can you still see the slides?
No, no, sir.
No.
No or yes?
No.
No.
Okay. Oh, don't you love this stuff? Share. Where is share? Share screen. All right, here we go. Can you see me now?
Yes, we got you.
All right, this is a little mussel called deer toe. It's about the size of a hickory nut, and it's sitting in an aquarium, and it's in the mood, and it's releasing thousands of particulates into the water. Well, millions of particulates into the water. These are not individual sperm. Individual sperm are much too small for you to see, but these are aggregates of sperm called spermatozoomata, or colloquially, we call them sperm balls. And this mussel will keep this up for a day and a night. It's really impressive. They've got amazing stamina. Okay, now here's what those sperm balls look like.
Oh, wow.
And these are the individual sperm. Now, we've added a little bit of sodium chloride to the water. Remember I said salt is an important pollutant? Sodium chloride makes the sperm balls disperse. And we think this is an abnormal example of a normal response. I mean, they're supposed to disperse. The sperm are supposed to come off the sperm balls, but only when the sperm ball is inside of a female mussel. How does it get there? Well, they drift downstream. The female presumably filters them into her mantle cavity. Then they disperse. The individual sperm come loose and enter her gills and fertilize the eggs. That's the story. But sodium chloride will trigger this outside the female if the concentration is sufficient. So this is one way that a simple mineral salt can interfere with mussel reproduction. Yeah, the sperm balls are really interesting. And let's see if I can, here's another video that shows that. This is a higher magnification of a sperm ball that's about 6 hours old, and it can swim. It's like another multicellular life stage, almost like a ciliated protozoan. And they probably exhibit some taxis, not to find the female, but to stay up off the bottom because they're fairly dense. And if they didn't swim, they would just settle out. But again, if you add salt to these, which I'm about to do, it will trigger the dispersal, and you'll see these going off like little fireworks. So that's how that works.
Awesome. Thank you, Chris. I think we had a question on chat about zebra mussels in Missouri.
Yeah, we've got zebra mussels in Missouri, but they haven't become a huge issue, not nearly as huge as we expected them to be. They actually showed up in the early 90s in Missouri, and they're fairly abundant in some of the small lakes in the Kansas City area. We've done a little bit of work with them at Lake Lotawana, which is a little residential lake, and they've become more of an issue in Kansas. I think they're in bull shoals. They become very abundant in bull shoals, but I don't hear many complaints about them. You know, the conservation department had billboards up for years saying, "Don't move zebra mussels." Well, you know, it was always just a delaying action. They were bound to get dispersed eventually because they're very good at hitching a ride in boats and live wells and so on.
Can you
And
elaborate? Oh, sorry.
yeah, so I'm just meandering here.
I was gonna ask if you could elaborate on their, native ecosystem impacts. I know you showed they can gum up, native mussels, but what
Yeah,
what
their
are the
economic
consequences?
impact. Their
Yeah,
economic impact has more to do with pipes and filters and screens.
no, I was wondering about the ecological impact. What impact do they have on, native species in those communities?
Yeah, they're very effective at filtering the water. So they're in competition with other things that filter the water. And in cases where they become super abundant, they can depress the food value of the water to the point where they affect the growth and population density of native animals. And this has happened particularly in
Great Lakes.
larger rivers. Let's see, I lost somebody there. But here in Missouri, you know, I'm not aware of any studies of ecological impacts in Missouri because, frankly, most places in Missouri, most of the rivers, they just haven't gotten established. They've only become a huge issue in some reservoirs and lakes. That's where they become abundant. Bull shoals is loaded with them, but their populations sort of crest and then they crash, sometimes for reasons that aren't entirely understood. And you know, it's just like all other invasive species. I think eventually they're going to come into some kind of a balance. Predators and parasites and disease are going to take advantage of this resource that's available. And, you know, I think we're already seeing that in some places.
We've got a question online. Can you talk about how conservation is being addressed and what organizations might be taking this up?
Yeah, well, that's a good question. The Fish and Wildlife Service, of course, is responsible for the endangered species. And the Columbia Office of Fish and Wildlife Service has a couple of excellent malacologists, both of whom were graduate students of mine. And that's Andy Roberts and Brian Simmons. The Conservation Department has a malacologist, we have a state malacologist, that's Steve McMurray. And Steve and Scott Feynman, another Conservation Department employee, have done a lot of surveys around the state. Their main activity is monitoring populations and trying to identify problem spots and interfacing with Endangered Species Act and the feds. Because if a government agency, say MoDOT, wants to build a bridge over a river, or if a farmer wants to revet a bank, or a pipeline is going across, or something of this sort, you have to find out if there are endangered species that are going to be impacted by those actions. And the way that's done is by Conservation Department and or Fish and Wildlife Service taking a look and determining whether a survey needs to be made and what sorts of problems might arise. So those are the go-to agencies. And then, of course, on the research side, universities, including mine, and government agencies like the Columbia Toxicology Lab that I described, very important in mussel research, not just for Missouri, but nationwide.
Well, thank you. Any questions in the room? Over here? Okay.
Two questions, Chris. First question is over at Bush Wildlife, we were there recently on a birding trip of all things. And we saw the lakes kind of drying up. And there were lots of mussels, which our resident birder, David Betcher, thinks were being eaten by limpkins. Okay. What I'd like to know is if a lake dries up, can mussels survive under the mud during that drying period? So when the lake fills back up, the lake will be replenished with mussels, or are they gone?
Yeah, immersion, EMERSION, immersion is an issue. I mean, these are aquatic animals and they do have some ability to survive out of water. And it depends a lot on the conditions. If the temperatures are moderate and if it's cloudy and, things stay wet, they can go on for a very long time in some cases. There are species that can last over a year out of water. Now, those species are typically living in habitats where they can expect to be immersed, like losing streams in the western parts of their habitat range. And even typical river species can typically last for weeks or even a month in very mild conditions. But if it's freezing cold or scorching sun and bone dry, mussels die, and they die in huge numbers. And this is it's part and parcel of the disturbance regime that rivers have been undergoing for the last couple of 100 years. Channelization, siltation, impoundment, and
Are there any
immersion is part of that. It's another disturbance that mussels have suffered from.
Are there
Is
any
it worth
methods?
throwing them back? I'd say, yeah, it is worth throwing them back. If you have a low stand of a river and you see a lot of mussels that are clearly alive, throw them back in the water. We've done that many, many times over the years. It's a feel-good thing to put mussels.
Are there any methods or sort of life stages that can withhold longer maybe? Or do they, if it dries for a long period of time and those species are gone, do they just have to be recruited again?
They'd have to be recruited again. Yeah, I wouldn't say that the juveniles are any more resistant than the adults. Typically, it's the adults that are the most resistant.
Okay, second question then. This has to do with Lower Merrimack River as it's approaching the Mississippi. I've walked along the banks there at times in the past, and you can just see hundreds of mussels on the shore, dead mussels out all over the shore. And is that a sign of mass die-off, or is that just the mussels are dead and there's still a lot of mussels still in the river? Is this a kind of a symptom of pollution where you see lots of this, or is this just part of the natural process?
Bill, I hate to say it, but you're going to have to repeat that one for me.
Okay, so the question is I'm walking on.
Yeah,
I'm walking on
mic's not doing the
shore.
job.
Okay, I'm walking along the shore of the Merrimack River near the Mississippi, so on the lower Merrimack, and I see lots and lots of very large dead mussels on the shore. And I want to know if that's a natural process where that's just the animals are dying and that's where they end up, or is this a pollution process or a disturbance process where now the river in that stretch is gone for all the mussels that should be there? Is that a sign of them being wiped out, or is that just normal life cycles and what's left of them after they're done?
Okay, I got you. It's not easy to tell is the answer.
Okay.
It could be a natural accumulation. Mussel shells do accumulate. Like I said, they last for centuries. You can find mussel shells that are hundreds, if not thousands of years old, eroding out of riverbanks, and they're still articulated. You can identify them as ancient shells because the nacre isn't glossy anymore, it's chalky. But it's not unusual to find mussel shells in an area where mussels are thriving. On the other hand, you can have mussel beds where there's tons of adults and hardly a shell to be seen. It all depends on the hydrodynamics, where shells accumulate, and that depends on how the river is flowing and what the channel structure is like. So it's not an easy question you're asking. if you're an optimist, just assume that it's a good sign that mussels are out there. But one thing you could do is you can look at the shells, and if there's tissue in there, then you got a problem because that mussel died, within a month or less, if there's still tissue in there.
Thank you.
Sometimes if they desiccate in the sun, the tissue might be in there for over a year, but fairly shortly, it'll be an empty shell. And, it didn't die recently then.
Okay.
So that's a good thing. But if you find a lot of mussels that are freshly dead, then you've definitely got a problem.
Good to know. Thank you.
We had a question
It's
about.
it's a wonderful thing to find a bunch of fresh mussel shells. If you're not, if it's not a disaster, if it's just like a muskrat midden, it's delightful. Of course, there are rules about collecting now. You got to be careful about that. Be sure you read the wildlife code, or don't tell me about your mussel shell collection.
What about collecting empty shells? Is that potentially problematic?
There is a rule, and you'll have to read the current version because I'm not sure that I've read it in the last few years, but the rule used to be that you could have a small number of mussel shells in your possession. And I don't know if that's the case. I should have checked that out before this talk, but I've been working on butterflies for the last few years. So,
Right.
and by the way, if you guys should want a return engagement, I'll be happy to talk to you about our butterfly work sometime too.
I think that's a fantastic idea. Hey, we had a question about what potential impacts there are from dredging river channels on mussels.
It can be very bad. Yeah, dredging is a big problem. I have some experience with this from the Missouri boot heel. This actually became a bone of contention, but you may be aware that the waterways in the agricultural part of the Missouri boot heel are really, they've been turned into ditches. They've been laser lined and they're excavated to a certain depth, and they're an amazing feat of engineering in what was just a meandering swampland. Now, you might think that that would be sterile and dead, but there's a lot of mussels in those ditches, and they're there because the engineering has made those ditches very stable habitats. Now, to keep them as stable habitats, they have to go in every couple of decades and dredge them out. And because rivers don't want to be straight, they want to meander, and they do. Well, that destroys a lot of mussels. There's about 10 or 11 species there that are quite abundant, and they do come back. But the act of dredging destroys them, and at least one of them is a threatened species in some places, the fat pocketbook mussel. So, the habitat is an artificial habitat that's been created by dredging. And the maintenance of that habitat destroys quite a few mussels periodically and in certain places. But overall, you could argue that, que sera, what will be, will be. And there's some kind of stability, at least, and that the mussels do come back.
Oh, thank you. Pete?
Are there any Missouri species that have become more abundant?
The question was, are there any Missouri species that have become more abundant, I guess, lately?
Oh, yes, no. Unless you want to count zebra mussels, which are not native mussels. But no, I have not seen any sign. There are some places that have gotten better. In one happy case because of protection, and that's the Verdigris River Mussel Refuge in eastern Kansas. That's a fantastic spot that was over-harvested commercially at one time. And it's been monitored for decades by Ed Miller of Kansas Wildlife and Parks. And it's in great shape, and it's gotten better. Why? Hard to say. Protection of the habitat, I guess. Maybe some relief from commercial harvest pressure had something to do with that. But I guess I'm getting old and cynical. No, I just can't think of a place in Missouri where things overall have gotten better.
Right. And I guess it's safe to assume the sort of the center of diversity for this group is what, in the southern half of the state, the Ozarks, or would it be more towards the southeast like the Boot Heel?
The best place for mussels? Is that the question?
The best place for their diversity in this state,
Merrimack River.
Merrimack River.
Yeah, the Merrimack is the beating heart of musselsdom. And get out there and look at them. There's still some great places. Pacific Palisades is a great place to look for mussels.
Interesting. Thank you. And I had a question on, you said, freshwater mussels are harvested to progenate marine clams or marine pearls.
Yeah,
With all the marine species, what makes that so versus, breaking up pieces of marine shells, for instance?
yeah, this is a highly developed industry. And there's a couple of places where historically it's been very important. One of those is in Japan, but the ones that come to mind are in the South Pacific. And the species that's used there is a big flat bivalve called Punctata margaritifera, and it's huge. It's as big as a washboard mussel. And those are nucleated with beads of shell drawn primarily from washboard mussels, from the freshwater mussels, because washboard is our biggest mussel and has the thickest shell. And that means that you can make the largest bead, which gets you the largest pearl. And there's a nice video online by Palisby Pearls about their industry. It's a highly developed industry and it's big bucks. And they farm the pearl oysters. They farm them, and it's a remarkable industry. But the freshwater trade is more a mom and pop operation in China. And you might have a subsistence farmer who is on a waterway, and he might, from a dealer, buy a bunch of nucleated freshwater mussels. When I say nucleated, I mean that they've been operated on. And in the case of the freshwater mussels, they don't use a bead. They just sacrifice a mussel, take the nacre secreting, the mother of pearl secreting tissue from that mussel, cut it up into little pieces, and then tuck those into the mantle of the pearl making mussel and put it back in the water. And that mussel grows, and you might have at harvest a dozen pearls on each side. And they look like blisters under the tissue that lines the shell, and you just pop them out with your fingers. It's incredible. And they can be manufactured, they can be grown in such quantity that the prices of, they're not comparable to the marine pearls at all. I mean, something like this used to be only royalty could afford pearls. If you look at paintings from the 17th century, that's what Columbus brought back to the Queen was pearls. That was extremely valuable, but now it's like a few bucks because it's kind of manufactured.
Yeah, I think we had a related question online. Someone asked, what methods were used to harvest mussels for the button industry?
Well, that's a good question. I've got a video of that. Are you interested?
Sure.
Do we dare start a quick video? I think it's 2.5 minutes.
I think we've got enough time left.
See if I can find it. All right, can you see my screen?
Yes.
All right, this is the clam diggers. Now this is probably the last ditch of commercial button shell harvest in North America. This was on the Grand River in Ontario in the early 40s. I got this from a guy that works for the Canadian Federal Conservation Agency. And these guys, it's just a family group, it looks like, and they're out collecting shells. But this is how abundant mussels were. This guy is just reaching down. He's not even looking. He's just reaching down, picking up mussels, chucking them into his little barge there. One after another after another. I've been in a few places where mussels were that abundant, but not many and not of any size. So these guys, they're just doing this on the weekend. This is not a life's pursuit here. This is more like a hobby. But the thing that's striking is just how incredibly abundant the animals are. And now he's got a pitchfork and he's shoveling them into a big bin there. I'm going to jump ahead a little bit. Now they're boiling them out. This is how you kill the animal. The shell opens and the meat falls out after it's been cooked enough. Look at the pile of shells these guys have, these weekend warriors. I mean, it's tragic, but it's incredible too. Now he's just chucking the mud out of his little barge. Yay. Mussels, get it? It's like the buffalo. It's like those piles of buffalo skulls on the prairie. Well, that's how it was in all the rivers of Eastern North America. That's how it used to be. And that harvest, it's just not sustainable. And I'm sure they'll cart those off to a factory nearby. This is the end of the industry, just before the fishery ran out and people realized that plastics were cheaper and in a lot of ways more suitable as button material. But if you've got a dress shirt, take a look at the buttons. Your buttons are plastic, but they look like mussel nacre. That tradition has persisted this long. Even though shell buttons haven't been manufactured since the 40s, we still buy shirts for dress that have what look like pearl buttons. Okay, that's enough of that.
Okay, thank you.
There were other ways that they were harvested. It wasn't always hand picking and pitchforks. There were structures called braille bars, which are bars that had chains hanging down, and then there were what were called crowfoot hooks, which is just little clusters of wire that when this was drug along the bottom, it would fall between the two valves of the mussel, and the mussel would clamp onto it and then get pulled up. And brailing was a popular way of harvesting shells when they were still abundant. So, Bob, I hope that answers your question.
Thank you. Yes.
Okay, any other questions from the room? Okay, any other questions online?
Hi, this is Phil Koenig.
Hey,
I
Phil.
almost got to visit you in Springfield with Casey, but I missed out on the opportunity. But I'd love to hear you talk about fritillaries and the Atala.
Well, we'll do that. We'll do that sometime this year.
Great.
If you guys can fit me into your schedule. Is Casey there? I can't recall. Casey was going to make it.
Casey's here. We're gonna come down and visit in May and June.
He's the one that sets the schedule. I'll be there.
All right.
All right, if that's all the questions we have, let's